So now we have our last speaker, making a scientific presentation, Neill Liptrott from the University of Liverpool. So Neill, 11 minutes?
I'll aim for 13.
Okay.
Okay. Excuse me, I've got a cold, so if I cough through this, I apologise. So, as Patrick mentioned, I'm based at the University of Liverpool. And hopefully the slides will move. There we go. Let me skip back one. There we go. So I'm based at the University of Liverpool. I'm a lecturer in pharmacology, and I'm the assay group leader for the immuno and hematotox characterization as part of EUNCL. So to follow on from Sven Evans' talk, what I want to try and show you is a little bit of data that we've generated on this liposomal irinotecan formulation that Sven Evans was talking about in his presentation. So to skip through this relatively quickly, I think everyone here is pretty much aware that there is a need to examine the interaction of nanomaterials and nanoparticles with the immune system. Unfortunately, I don't have time to go, well, I'd like to, I don't have time to go into all those different interactions because I'm an immunologist and I can quite quickly put people to sleep with most of my talks on that kind of thing. But we do know that there are things like immune stimulation, immune suppression, different blood contact properties that have been demonstrated to be possible with nanomaterials and nanoparticles. Immunotoxicity is not something that's new. It's already part of the evaluation process. But both the EMA and the FDA have various documents and guidelines out there. There's even now the specific ISO 10993 for immunotox testing in medical devices. So it's not a new process. It's not something new. But one of the things that we try and do on the nano side of things is make those assays relevant for the materials that we're going to test. Although there's a lot of work out there on structure-activity relationships and trend analysis, and that's something that we're doing as part of EUNCL, as to what physicochemical characteristic actually may cause these issues, at least to my understanding and to my knowledge, there's no definitive link between certain processes at the moment for different materials, but obviously there are some strong associations there. Some considerations for the preparation of the particles. So batch-to-batch variability is something that's very important in general, but also for EUNCL. So one of the things that we like to try and do is try and at least compare two batches if they can be produced by the sponsor. Endotoxin and pyrogen contamination is obviously vital to the immunotox cascade to make sure that we don't get any false positives in any of the assays. And also chemical contamination is important as well because we want to make sure, as Sven Evans was saying, you know, if we do see some cytotoxicity, we do see some of the cells dying, we want to try and make sure whether it's not because of some contaminant that's left over from the formulation process or if it's the actual materials themselves. So, on the immuno and hematotox side of things, I've used this image has been adapted from a paper that Marina published. So Marina is the immunology lead at the NCL in Frederick. And Marina published this paper suggesting a tiered approach towards immunotox testing for nanomaterials. And so I've both adapted the figure, but also we've adopted this kind of process at Liverpool for the materials that either we're developing in-house or that we work with through either EUNCL or other partners. So the first thing we do is we have our pre-screen, which EUNCL does. We do endotoxin and sterility testing to make sure that there aren't anything in there that's going to cause any problems or false positives in the subsequent assays. Then we tend to start off with the more common acute toxicities, things like hemolysis, complement activation, and cytokine secretion. Depending on what we see within that tier, we then tend to go through into more sort of functional assays where we look at things like macrophage function, so phagocytosis, cytokine secretion, and chemokine secretion from macrophages. We can look at NK cell cytotoxicity. You know, the list goes on. There's quite a large potential battery of assays that we can look at. And then to try and tie it all back together, we'll then sort of try and work out how some of these physicochemical characteristics link into some of the differences that we're seeing, some of the effects that we see in the cells that we do. And what I'd like to do is just highlight that we have a few posters here from some of our more mechanistic assays that I know it's we're on the last day, but you may have already hopefully seen those posters and some of the mechanistic work that we've been doing. One of the things, though, that we always have to try and bear in mind, and this is something that's particularly important, let's say, for us at Liverpool, because our primary interest is in infectious disease and HIV, is that when we develop these materials, when they're going to actually be used in clinically, they're not going to go necessarily into, well, obviously they will go into healthy volunteers in clinical trials, but their intended use is in patients who may have a slightly different or altered immune system. So obviously from the context of HIV, there is this sort of basal level of information. So whether or not we see the same responses between the healthy volunteers that we use in our general assays in patients is something that we always have to try and bear in mind. So, some considerations from the assessment of the toxicity. I'm not going to talk about the cytotoxicity assays, that's a different assay group. But some of the considerations that we have for toxicity and biocompatibility materials, I've listed a few here. The list probably goes on, I've missed probably quite a few. But what I wanted to try and get across for the purpose of this slide is how EUNCL is approaching some of these issues using both our tiered cascade and the different assay teams. So what I'm very thankful for and I'm very lucky for, and most people don't necessarily have, but as Patrick was describing, this is one of the purposes of EUNCL, to provide these resources, to provide this ability of characterization. What I'm very lucky at is the fact that before any of these materials come to me and I start bleeding people to do these tests, they've gone through the physicochemical team and pre-screen. So I know by that point, with the work of Fanny and Luigi and Dora, I know that they're well dispersed in the different buffers that we're going to use, what conditions we should use to reconstitute them. So we've got a very well worked up SOP for how to just handle the materials before we actually start putting them into healthy volunteer samples. Ryan Rossig, who's based in Munster, he leads the endotoxin and sterility team. So as well as having that physical and that physical characterization of the materials, we also know whether or not there are any sort of endotoxin or pyrogens in there. The next thing we've got is this release profile in complex media, something that Sven Evan talked about quite extensively as part of the chemical characterization team. So we know whether or not in different medias there's going to just be a dump of the drug that's loaded with them, or if it's going to be released slowly, or possibly whether it releases the drug at all in the different medias that we're going to use, so we get some idea of stability. Linked in with the chemical characterization team is this idea of chemical contamination, whether or not anything's been left over from in the formulation process. The cytotox team are very useful there, and Suzanne mentioned some of the assays that they have at the JRC and both at Syntef, where we look at that to see whether or not we can get some early readouts if there are some chemical contaminants in there that might actually be causing any issues. For the immunohematotox team, I mean, these are not exclusive to that particular the immunotox team, but we always try and use concentrations that reflect those realistic nanoparticle concentrations. So when we work with a sponsor, if they have that in vivo data already, if they have some PK data, we'll base our assays and our experiments on that information that's available as far as possible. If not, we can use sort of different FDA scaling calculations to try and get an idea of what we class as the theoretical plasma concentration and work from there. But obviously what's very important for all the assay teams, and as Suzanne mentioned before, this interference with assays is something that we have to be very careful of. And the lessons that we learned very quickly from our work with the US NCL with respect to having the appropriate inhibition enhancement controls in the assays that we do. And hopefully I'll show you some examples of these things from our work on the liposomal irinotecan formulation that Sven Evan mentioned. So, as I say, it was we termed it EUNCL 11. It's a liposomal formulation of irinotecan. Following all the physical and chemical characterization that Sven Evan mentioned that was carried out by both CEA, Syntef and JRC, and through pre-screen with Reiner at Munster with respect to bacterial endotoxin contamination, it then passed through into the immunotox group. And these are the assays that we listed from our, it's available on our website, all the SOPs. These are the assays that we applied to it. But what I want to try and do is show you some examples of these specific assays for different reasons, hopefully which will become apparent. So not just to show you our capabilities, but also to show you why we might prioritize certain assays over others. So this work was conducted in parallel with both Martin Brudder at Oxprotect and Tobias Gerdes at VivoScience. And as well, we used this characterization campaign to harmonize our assays with the NCL in Frederick as well. So both the US and Europe conducted similar, our respective assays on this material, and we compared results. So what we did find was that EUNCL 11 didn't cause any hemolysis in healthy blood cells, in healthy volunteer blood, I should say. This is just some data to show you that we can generate hemolysis using our positive control, which is just a detergent, Triton X. There are these accepted limits on what is or isn't hemolytic. Anything less than 2%.
We will have at 11:30 the big plenary session on cancer with Professor Patrick Couvreur and with Professor André Neill. We invite you to go now to the Hall Montreal.
So thanks very much to Bert for his contribution to my talk. All questions can be addressed to him in the hall outside. So I'll take another 20 minutes if that's all right, Patrick. So this is just to show we can measure hemolysis in whole blood. Now, what was interesting from this with respect to possible interference with assays is when we initially conducted this work, as I say, anything less than 2% we would say isn't hemolytic, 2 to 5% is slightly hemolytic, and anything above 5% would constitute a problem with the hemolysis. So when we did this work initially and we used these 4 different concentrations of the material, initially it looked as though there was some hemolysis that had actually been generated. However, when we then compared that with our controls, with the controls that we use in the assay and normalized it against that, there was effectively no hemolysis whatsoever. It was just interactions of the materials with the assay that we tested. So that's just to give you an example of how we use those controls to test these different things. When we looked at complement activation, we saw no complement activation with all the concentrations that we tested. Our positive control is cobra venom factor, and again, this was done between three separate labs for a comparative analysis. We saw no complement activation right up to 60 times what we classed as the theoretical plasma concentration. So we try and push toxicity in these assays as far as possible to try and get an idea of what will actually cause any issues. And we went as far as possible with respect to solubility of the material. We went up to 60 times the concentration, and we didn't see any activation. When we looked at leukocyte proliferation in peripheral blood mononuclear cells, so this is just some data to show different concentrations and of the positive control that we use. So we use a mitogen here, we use PHA, and you see as they increase the concentration, we see more proliferation. What I wanted to use this slide was to show you the variability or the possible inter-individual variability we see between 3 donors. So there's 3 donors here represented by the different colours, and you can see that they all respond quite slightly differently to that mitogen. That's important for something I'll mention in the next couple of slides. So this is just the percentage of the untreated control. So this is just the materials or the cells exposed to the materials in and of themselves. And what we did see is as we increase the concentration, we see much less proliferation. So there's obviously some form of cell death going on there as we treat the materials. What we also do is we co-incubate those materials with our positive control to see whether or not they enhance or inhibit that response. And we saw the same, we saw the same thing there, where we saw that as we increase the concentration, we see that there's an impact on proliferation. Now, why might that be? Sven Eben already mentioned it. It might be due to the accumulation of Irinotecan and or SN38. Now, previous work that we've done at Liverpool, we know that the proteins involved in drug disposition and drug distribution are expressed and active in immune cells for different reasons. It's also stated that the UGT1A1 genotypes and polymorphisms, as Sven Eben mentioned, it's UGT enzymes that glucuronidate the SN38 and effectively inactivate it. And variability in some of those genes might be why we see this impact and this variability and impact in these systems. But what we're going to do is follow this up with the chemical characterization team to see whether or not we see the presence of the different SN38 and the glucuronide in the cells and the samples that we test. Thanks to Bert. I've run out of time, but what I would say is that these materials have now been passed into the in vivo assay group. So the in vivo group is led by Adriele Prinamello at Syntef, and in context of that, in collaboration with Sven Evan at Syntef, there's an ongoing in vivo experiment where we're looking at or I should say Adrieli and his team are looking at the in vivo PK and tissue distribution of the formulation using both 2D images and 3D reconstructions of the 2D images. And as well as that, Sven Evan, who I'm definitely not a chemist, can give you more information on the mass spec quantification that they're using there. So very quickly, hopefully you've got an idea from the session that we can provide this robust, relatively smart assay cascade, interconnectivity of these assays to try and make it more relevant for the things that we do. For now, we treat each material as unique, although we will prioritize certain assays based on those particle characteristics. And for me, important, the last two are what the real take-home message. The analysis of the particle characteristics in different matrices, as Sven Evan stressed, and whether or not there's any biological or chemical contamination is vital for the immunotox assessments. And with that, I'll leave you with acknowledgements both from my group and the US and EU collaborators, and thank you very much for your time.